Hello and welcome to Brainstorm Webinar Week. This is the second part of our four-part Brainstorm webinar series. And today we're going to focus on dSTORM in primary cells. Yesterday we had a session on the introduction of dSTORM, and tomorrow we're going to continue on extracellular vesicles and dSTORM, and ending this webinar series with analysis. Before we get started, I want to mention a few things, a few rules for this webinar. So you will see that on this webinar platform, you have a little panel and two important things to note. You have the chat function, which is important for us to receive as many questions from you, from the audience. And you also have the handout section where we have left a few resources that hopefully are useful for you information. Also, this webinar is going to be around 45 to 60 minutes, and it will be recorded for later reference, and you will find it available in the future on our website, oni.bio. And don't forget, if you are missing any resources that are not in this handout we're providing, you can find more information in the resource section of our website, oni.bio. And with that, I want to welcome you all. Thank you for joining. And we're going to start with a little introduction of our speakers today. So my name is Laura, and I'm a field application scientist for ONI. You may know me from conferences, from training and installations, and everything around customer support in the European area. And with me today, we have Stefan. Please, Stefan, can you introduce yourself?
Thank you, Laura. Hi, everybody. So my name is Stefan. I'm a senior application development scientist at ONI. So after earning my PhD in biophysics, I moved to pursue my career in single molecule localization microscopy, where I focus on the development of the correlative live cell imaging methods based on single particle tracking and high resolution microscopy. Before joining ONI, I used my knowledge in super-resolution microscopy to answer open questions in immunology and biology. And by using multicolor dSTORM imaging, I discovered the supramolecular attack particles, or SMAPs, which are tactile arsenals of T cells secreted to the immune synapse.
Thank you, Stefan. I hope we're going to hear more about your experience and research. And actually, let's go to the agenda of today. So today we're going to discuss a few things. We're going to start with a little recap of the basics. We're going to discuss dSTORM in cell lines and how does it compare with primary cells. And we're going to go a little bit deeper into strategies for primary cells labeling, as well as how can we image precious samples and how can we make sure our experiment will be successful. Before we begin, we have a short poll for you to get to know our audience a little bit better. So let's take a quick questionnaire. Our question today is, where are you now on your microscopy journey? We have three options. Maybe you have no experience, that's fine. We are here to learn. Maybe you have already some experience, or you are an advanced microscopy user. We will give it a few seconds for you to answer, and then we will review the answers. I'm very curious what our audience is like today. Amazing. So I see we have most of the audience today has some experience with microscopy, and close together we have also a lot of people that have advanced experience. Fantastic.
Okay, so that actually sounds really good. So, but as we start, I would like to just give you a small recap, basically about the basics if you didn't attended it yesterday. So then we just give you a small introduction to single molecule localization microscopy, and we will start from there after. Okay, so single molecule localization microscopy. So as it already says in the title, it's looking for localizing single molecules in your samples. So as you showed, you are more like 50% of you has a high experience in high resolution microscopy or in advanced microscopy techniques. So you know that the diffraction limit where we can separate two objects from each other is currently approximately around 200 nanometers. But a lot of proteins, a lot of molecules are actually working in a much smaller scale. So, and that's why where the single molecule localization microscopy comes in. So this is a technology which is actually utilizing individual fluorophores by using high laser power. So as you can see here in the video presenting. So if you use high laser powers, you can actually switch individual molecules to a on or off state. And as you see, they appear like a small dots. And by fitting each individual dots, you can actually resolve the high resolution image. So suddenly you can actually achieve a resolution somewhere below 20 nanometers. So this is a basic of the single molecule localization microscopy.
And Stefan, can you just briefly mention why, in what scenarios on cell imaging, why is this gain in resolution very powerful?
Yeah, so this is very interesting when you are looking for primary cell, when you're looking on your cells, when you're looking for protein-protein interactions. I will give you just an example from literature or from personal experience. We are looking, for example, for a mitochondria. Mitochondria has an outer membrane, but also the inner membrane. And there are some proteins which are actually associated with. For example, TOM20 is a very common stain for the outer membrane of the mitochondria. But then you can have a TIM44, which is a stain for a inner membrane. If you will use a normal conventional microscopy techniques without breaking the diffraction limit, you will actually get these two colors overlap. So you will not see a difference. You will not see whether your label is actually labeling the inner or outer membrane of mitochondria. But this is when these microscopy techniques come to place and gives you a high advantage. Because when you will switch on individual fluorophores from each of these two colors what you will use, you'll be able to basically resolve the separation between the inner and the outer membrane of the mitochondria. And that's where these technologies are really useful. They can look on a protein-protein interactions. In primary cells can look between a T cell receptor and a CD3 or any other FC receptors and so on. And there are multiple publications showing that a lot of these interactions are really happening at the nanometer scale. That's where these technologies are really, really powerful technology.
Thank you for this very interesting example. And I want to make sure to stop for 1 second to ask if there are any questions about the basics of single molecule localization microscopy or dSTORM. Since yesterday we covered most of the part, we are going to keep the sections short and dive right into application afterwards. So are there any questions about SMLM at the moment? If not, don't be shy and just leave us your questions in the chat and we will make sure to incorporate it in our conversations. So our next, we're going to dive right into the application in our next section, which will be focused on imaging cell lines versus primary cells. And for that, we have prepared a second questionnaire for you to understand a little bit more about actually your experiments and your experience. So our question for you is, what cell types are you currently using? Are you using mostly primary cells, or are you using cell lines, or are you saying, no, I'm actually using tissue, I'm very interested in tissue imaging? What cell types are you using at the moment? Also, we will give it a few seconds again, so you can answer. All right, I'm very curious to hear. Oh, we have a nice mix with most of the participants today using cell lines. And then we have 15% of users using primary cells and tissue imaging, which, to be very fair, are also the more difficult or tricky samples. So let's dive right into the cell imaging part. So my first question for you, Stefan, is a little bit more related, like how does a general cell staining protocol vary if we do high resolution dSTORM compared to other traditional immunolabeling with confocal? Is there a major difference between these two?
Yeah, so this is a very interesting question. Basically, the answer in this will be no. So if you already are using actually some conventional microscopy techniques, you are really free to be able to use this. However, you need to take into account some very important things. One of the more important things, the most important thing is that dSTORM or single point localization microscopy is imaged in turf mode, which is a total internal reflection microscopy. In other words, it's very close to the cover slip. So one of the main requirements for this technology is that you are actually using cells which are adherent to the surface, which sometimes might be very tricky when you're using primary cells, especially in immunology, as those cells are not adherent cells, but they are cells in suspension. I will later cover a little bit about it, what are the best strategies to use it. But overall, if you look from a labeling point of view, if you already are performing some fluorescent imaging by using confocal microscopy, you can freely use the same labeling strategies for single molecular localization microscopy. What you need to pay attention is to the selection of fluorescent dyes. Some fluorophores, for example, like PE, is like very commonly used in FACS or flow cytometry, and when you scan it, you get one shot. It's very rarely used in microscopy, but sometimes in confocal microscopy is actually used. However, for single molecular localization microscopy, that dye is not suitable. So you need to have there's a dye selection list, which are dependent on your imaging buffer, what you're using in technology. But overall, there's not really a different if you are trying to do a different strategy to your confocal imaging, to your labeling of your sample, as for example, for confocal microscopy.
And does that, so if I'm hearing correctly, is that it's more important to watch out for the fluorophore instead of?
Yeah. Yes, exactly. So the fluorophore. So we actually are, because in single molecule microscopy, you need to switch on and off this fluorophore by using high laser powers. So not each fluorophore will actually switch on and off. You most probably have the experience when you're a confocal, you increase the laser power and suddenly everything bleaches. So this is exactly what you guys would like to avoid. So the best thing is you need to make sure that when a high laser power is on, your molecule is only switching on and off, but it's not actually bleaching. At least not for until you are able to resolve your image. So that's why there's a list of fluorophores which are used for single molecule localization microscopy with the specific buffers. And there's a multiple different buffers outside, and I would say in tens of different buffers. And for each buffer, there's a different fluorophore that works well.
Yes, and I believe we have some more guidance for anyone that's interested in the handout section, where we have some fluorophores for you for your reference. And what about the cell, the wells in which I have to seed my cells or add my cells to? What do I need to watch out? What wells can I use?
Yeah, so if you are using if you're using for microscopy techniques like for confocal microscopy, if you are using a high magnification objectives, you need to make sure that you are using cover slip maximum with the number 1. So they are not very thick. So ideally, it's a quartz cover slip and it's not very thick. Because your imaging depth or how deep you can actually image will depend on the numeric aperture or the working distance of your objective, which is the link to numeric aperture. So, like if you use a low magnification, then even you can use your plastic 6-well plates, what you are using to grow your cells in a tissue culture. because the working distance, or the penetration of the objective into the depth of the sample, is in a range of almost like centimeters. But when you increase the magnification and you want to look at an individual cell or individual organelles within a cell, then suddenly that working distance from the objective is much smaller. So this is when it's somewhere around 170 microns. So in this case, you really need to watch out that your glass cannot be basically thicker like 100 micron if you want to get deep in 70, because the thickness of the glass also matters. So ideally, for single microlocalization microscopy, you will use dishes which have a glass bottom. The thickness of the glass should be somewhere maximum to number 1, which is around those 100-120 microns. And but you can use any dish which has this requirement. Whether you are using dishes like 6-well plates, flow chambers, Petri dishes, be the glass bottom. So the most important part is a glass bottom dish.
So glass bottom it is, but what about surface coatings? Can I use PLL or collagen? Is that compatible?
Yes, so that's actually a good point, especially when we are looking from a point of view of primary cells, or even cell lines which are cells in suspension. So when I worked in the immunology field, you very commonly use immune cells, so T cells, NK cells. You can also use macrophages and dendritic cells. So in this case, if you are if someone familiar from immunology, when you isolate monocytes, monocytes can be then converted to be dendritic cells or macrophages. But macrophage and dendritic cells are growing on the surface. So these are ideal cells because at the end they become like adherent cells. So if you grow them on your Petri, on your dish, on a glass bottom dish, you don't really need to do any manipulation with them to be able to image them by single microlocalization microscopy, at least in terms of adherence to the cell. But then you come to a point when you're looking on T cells. T cells are cells in suspension, or cell lines, for example, like a Jurkat cell line. It's also a T cell line, but it's in a suspension. So in this case, for some, you will need to immobilize your cells to surface. So you can do activated or non-activated surfaces. For example, you can use polyalysine. Polyalysine will be negatively charged. It will attach your cells to the surface, incubating for like 10-15 minutes with your cells. Your cells will adhere to the surface, and they will not get activation. However, if you are interested to activate your T cell receptor, let's say the T cells, if you're talking about primary cells, then you can use antibody-coated surfaces. Usually it will be anti-CD3, which will then bind the CD3 molecule protein on the surface of the T cells, and that will adhere the molecules to the bottom, and then you can actually also use that as an activated surface in this case. So here you will look for a reorganization, for example, of your protein in the on activated surface, not on the non-activated. Another option is like using collagen. You can use collagen and you can use a different stiffness of it. But again, you need to pay a very big attention to the depth of your collagen. As I mentioned earlier, like if the working distance of the objective of around 100x objective is like 140, 170 microns, your collagen cannot be bigger like 30 micron maybe. And the reason is that your glass will be around 110, plus you add a 30 micron of the collagen, and then your cell is sitting on top of the collagen. So you can use it, or you can use fibronectin, but please keep in mind that the depth of your collagen or stiffness will matter, because that corresponds to your imaging. Also, please be careful that if you're using collagen, the material has a different density. So in this case, the light penetration is different. You can get the backscattered light is different. So when you're collecting the signal, you might collect a higher, like lower signal to noise ratio. So you will basically collecting a lot of noise. So you will need to be able to filter it out. So these are the options.
It's possible, but it can be a little bit tricky with the signal to noise ratio. We have to be very sure if that is maybe a thin layer of collagen or PLL.
Yeah, usually the thin layer of collagen or PLL is a right solution to go with here. But as I said, you can also use activated surfaces.
And so we have our first audience question regarding slides. And the question goes, aren't most microscopes optimized for number 1.5? slides or cover slips?
Yeah, so actually that will depends on your objective, basically, of your magnification. So as I mentioned earlier, like if you use any of the plastic dishes, what you culture yourself, and you go to look on those cells inside a tissue culture microscope, even if your cells are in a suspension, you'll be able to move up with the focus because your magnification around 5, 10, maximum 40 as an air objective. However, if you want to do imaging, you really need to go maximum to 1.5. A 1.5 cover slip can work if you are really looking on the bottom part of your cells. However, if you want to go more deeper inside the cell, let's say 5 micron thick cell, you get the macrophages, which are like big nucleus, and you want to go more in, then you might be on the edge of the detection of your system. Or if you are using slides for like histology, you can actually place your cells on those slides if you are interested in that. But then you need to put a small cover slip on the top of it. And then you will need to reverse your slide and image through the thinner cover slip. Those are some of the few tricks which you could do in this case. But ideally, I would recommend maximum the cover slip number 1 in this case. 1.5 could be an edge.
I see we're learning all the tricks from you today. Awesome. We have another audience question, this time from LinkedIn Live. Hello and welcome, LinkedIn audience. And the question is related to live cell imaging capabilities. And so I believe now we were talking mostly about fixed cell, but what are the limitations of dSTORM to visualize dynamic cell processes? Is that even
this is actually a very good question. There are some publications since 2006 when the technology was discovered that are using live cell imaging. As this technology needs a lot of localizations to resolve the image, which can lead to approximately 1,000 frames you need to record. Each of them in the fastest speed, you will go around 20 milliseconds. So, you know, if 20 times 1,000, you can 20,000, you immediately see that you need a lot of time to record it. A lot of objects are moving very fast. So if you are looking for a movement on a live cell imaging of like mitochondria, they are very slow moving organelles. But if you are looking for like vesicles, lysosomes, they are very fast. So in this scenario, you will not be able to use high resolution, at least not a single microlocalization microscopy to resolve the structures of these. However, there's an alternative solution for it. And that's that if you know that your processes in a cell are dynamic and change in time, you can fix your cells at a different time point. You can place your cells on a dish, fix them after a minute. You can fix them after a 5 minute in different well, after 10 minutes, half an hour, or depending where you see or where you expect to have some dynamics. And then you can do a labeling. And then you image multiple cells so that they will correspond to that specific time point. And that will give you the best resolution in this case.
Exactly. We also have to consider the high laser power. Sometimes we need dSTORM. So maybe other localization microscopy techniques would be more compatible, such as, for example, PALM.
Yes, you can use PALM, or you can use some of the DNA paint if you have molecules when you are actually using a double-stranded. The disadvantage of these technologies at some point that if you have a live cell, or you are encoding your fluorescent proteins inside, in your protein of interest, and then you have it expressed in your cell lines, or you do overexpression, or you need to look on the receptors outside of the cell, because you cannot use permeabilization. At least if you try to use permeabilization and try to label things inside the cell, you most probably will kill your cells. So, and that's exactly where we are coming to the point that if you are looking for a movement of receptors on a surface, yes, you could use some technologies like DNA paint or PALM, or actually, if inside the cell, you need to use PALM when you're expressing your fluorescent protein.
Talking about permeabilization, so permeabilization only for fixed cell. Don't permeabilize your live cells, please. Can I use permeabilization with dSTORM, or is there any concern that it will maybe damage my structure of interest?
Yeah, so the two main points here to consider. Before you go to a step of a parameterization, we need to talk about how we fix our cells, because this is one of the crucial steps which where you will decide which way to go. So we developed a kit which is giving you two options for fixing the cells. One is just using like a light fixative, like a PFA, paraformaldehyde, but the other one is a combination of paraformaldehyde and glutaraldehyde. Which of these two fixation is the best for your cells, you will need to actually try out. The reason why I'm saying is that by using any of this fixation, you can alter the epitope of your proteins, and then maybe your antibody will not bind to it. One of these actually very good examples is actually a T-cell receptor. There are antibodies which are actually for a T-cell receptor, but they don't really work when you are fixing your cells. And that's why people in alternative of T-cell receptors, they use CD3 or anti-CD3. And then you, in terms of permeabilization, as Laura was mentioning, yes, you can permeabilize. You can use a soft permeabilization method, which are reversible, such as saponin, or you can actually use, especially maybe if you're looking for the proteins in a membrane, so that you don't want to really open the pores in a membrane, but you want to open and they can close. Or if you are more interested inside the cells and get more deep inside, like a nucleus permeabilization, then you might need to use actually a Triton in this case. That's more harsher. It will open your pores inside the membrane. So that can affect at some point the reorganization of your receptors on the surface of the cell membrane.
Yeah, indeed. So a little bit of optimization is always necessary. And we have another LinkedIn Live question from Lisa from Florida. Welcome. So it's the question is, do photo bleaching dynamics, so does photo bleaching change? What is the difference in conventional fluorescence and in super resolution techniques such as dSTORM or DNA paint? How should we compare?
So photo bleaching is always basically happening, and it depends on your laser power and the presence of oxygen. So if you in a conventional microscopy techniques, you are using lower laser powers, which higher exposure times, which means that you are collecting the light from from your protein of interest for longer time, but you are not harming and you're not bleaching your sample. That's how you can prolong your imaging. In terms of single microcapillation microscopy, you are using special buffers, which are actually using, for example, glucose oxidase. And the whole role of the glucose oxidase is to get rid of the oxygen, which is causing you the photobleaching. So in this case, if you use like a STORM, when you have a fluorophore attached to a primary antibody or secondary antibody, you will need to use some of these buffers which have a glucose oxidase on it. I could also recommend you to use those in confocal imaging because they can help you. You will have less photobleaching, so you can image longer. But please don't really use them in a live cells because they are very harsh for live cells, and you will not have it. An alternative method is what Laura was saying, is to use DNA paint. In this case, actually, your protein of interest is bound to the strand, one strand of a DNA, and then you do a complementary strand which has a fluorophore. And how this technology actually works in a nutshell is that you have one DNA binding to another one, then the fluorophore is shining and you're detecting it, and then it's unbinding, and then you get another one coming in. So because all these fluorophores with the DNA stray are present in your sample, they will just recycling. So you will be able to image for longer, and in this case, you will also resolve a quite high resolution image.
Fantastic. So I want to dive a little bit deeper into the primary cells. So in our next section, staining strategies in primary cells, I am very curious to hear from you. What is the difference between dSTORM in cell culture cells and dSTORM in primary cells? What are the main things that I need to take extra attention?
Yeah, so this is a very interesting question because when you are looking from a point of view, we can say we use the cell lines, we are studying immune cells. When I'm studying immune cells, I would say every immunology lab will have a T cells, Jurkat cells in their incubator. So you can use these cell lines, but these are cell lines which are immortalized cell lines. We need to understand that the results from primary cells can vary from your cell lines, especially like if you consider a CD4 cell, like Jurkats are CD4 T cells, which should be considered as helper T cells, but they are also using for killing assays. But they should not be CD8 positive, so they are not CD8 positive, which in this case, they are good because they're immortalized and you can expand them, and they are giving you a group of cells which should always give you the same type of result. In terms of primary cells, you are getting them from different donors. So each donor is different. They can have a different HLAs, they can have a different ethnicity, and so on. And that will lead to a different subsets of T cells from them, for example. So in this case, if we talk about, or maybe go a little bit deep in it, like what type of results you could expect. So if you are looking from primary cells and you have a lot of primary cells and you expand them, you can optimize your staining on those primary cells. But don't be surprised if you take another donor and the results are slightly different. Really, don't be surprised. That's very commonly happening. That's why in this case, I would recommend to take at least 5 different donors if you are interested. In terms of cell lines, if you use a Jurkat cell lines and you do them the same treatment, you should always get the same result because they are like cloned pretty much.
And so I hear that primary cells are in that regard a little bit maybe heterogeneous is the right word.
Yeah.
Yeah.
Correct. So they are very heterogeneous. Yeah.
So how do I make sure that if I get samples, maybe patient samples, that I nail my experiments and my sample doesn't go to waste? How can I prepare a really good primary cell sample?
Yeah, so in this case, I would recommend first that if you are looking for any primary cells, try to figure out whether there is a cell line which corresponds to the same. For example, as I mentioned, T cells, they have a they have a Jurkat T cell lines or other T cell lines. Then you, as a target cells, you can use different cell macrophages, you have THP1 cell, TH1 cells. So first, I would optimize my staining on a cell line, especially when we are talking about primary cells when you don't have a lot. In terms of T cells, usually if you get the blood from a patient, you can isolate millions of these cells. CD4 is around 150 million easily. You can expand them, so you can play around with that. However, if you have a very precious sample coming it from a customer, like a patient, and you got a very small amount of T cells isolated from it, you don't want to waste them. What you do, you first optimize your staining with the cell line. You can take a Jurkat cell line, for example. If they don't express your protein, which you're looking for, you maybe express on it. Make sure that you are, because they're in suspension, you can use polylysine or collagen, that you make them adhere to the surface. Then you fix them after a certain time. You permeabilize or you don't permeabilize if you're interested on the outside membrane of the protein or the external part of your protein. And then you use antibody for staining. When you optimize that, and you are 100% happy with it using controls and all around, then I would use the same strategy for labeling on my pressure sample, on my sample which is coming from primary cells. If you have a lot of opportunity, a lot of cells from one patient, then feel free to make the optimization on those cells. But also don't, as I mentioned, don't be surprised if some of the results will vary between the donors, because you will really need to make a multiple repetitions with different donors.
I see. And one of my questions I had is regarding multicolor capabilities. So can I do multicolor in cell lines as well as primary cells? I'm going to combine it with a LinkedIn question. How do I make sure that my signal is very well separated when I do multicolor imaging?
Yeah, so that will actually depend on your imaging settings. So if you use for example, single microlocalization microscopy, and you are using, I don't know, ONI stanoimager, then we will have a nice separation, basically between the we have a separation between the 560, well, 488 and 560 channel and the 640 channel. So it's basically, I would say, like it's a dichroic mirror, so it's a split. In this case, you will see on one side of the camera only your one fluorophore at 640 and the other camera 560. However, in this case, you cannot do simultaneously the 488 and the 560 channel. But you can always perform it in sequential manner, and by using the right filter settings, you could be able to pick the right fluorophore. And that's why, as I said, there's a limited number of fluorophores, which in this case could be used, especially if you're using a high resolution mode, because you're using high laser power, so you can have a bleed through of some fluorophores to the other channel. So this is where it's a really careful selection of fluorophores with the imaging buffer for high resolution plays a big role.
And can you comment on what is the better labeling strategy, to use primary labeled antibodies or to use a primary and secondary antibody?
That actually will depend on your target, I would say. So also, if you are familiar with, let's say, main primary cells people with primary cells are mainly working in immunology. So if in immunology you already established imaging settings, for example, for flow cytometry, you can use the same antibodies and same type of strategy for a single molecule localization microscopy. You might just need to change your fluorophores. As I said, instead of PE, will not work for single molecule, but you can change it for other fluorophores which are compatible. We have a list of these fluorophores, I think, on our webpage, which work with our imaging buffer, because that's a very important thing. So if you have a protein, if you use a flow cytometry, you have your antibody conjugated to fluorophore, feel free to use that one. Use the approach of primary directly conjugated. If your protein is very low expressed in the cells, I would recommend to use secondary approach when you're using a secondary antibody as a primary antibody. And the reason behind that is that if you are using a conventional microscopy like confocal, you will make pretty much like one snapshot of it with a low power. But in high resolution, you need multiple localization to identify where your protein is. And that will be still within 20 nanometers. So if your protein is low expressed, you can enhance the number of localizations from that protein by using a secondary antibody, which has multiple fluorophores. And most of the time, you have multiple secondaries attaching to the same primary antibody. So this is actually, in this case, I would recommend to use an approach of a primary and secondary. Sometimes it's not possible, especially if you have same species. So it's very common, I know it from experience, it's very common that mainly you have a mouse antibodies, all IgG1s, and then suddenly how can I image? Can I image 3 mouse IgG1 antibodies? Yes, you can, but you need to have it directly conjugated to fluorophore. In this case, a secondary approach will just not work. So.
Yeah, thank you. So Darian from the audience actually has low abundant protein he's working with, or low abundant molecule, and is asking, how do I minimize the background noise then? I can imagine probably with primary and secondary, yes, you will most likely have a little bit more background noise. How do we minimize that?
Yeah, so you need to optimize your labeling strategy. So in this case, if you're using primary antibody directly conjugated, you always should use the control such as isotype control. So isotype control, again, the same species. So if you have your primary antibody is mouse IgG1, then you need to use an isotype control mouse IgG1 conjugated to the same fluorophore. When you do this, you will see what is your background level. And then you will see whether you need to improve in labeling, which means improving in blocking. So you can use BSA blocking, for example, BSA to block your surface better. If you get a minimal attachment from your isotype control, then you are lucky and things work well. So then feel free to use your primary antibody directly conjugated. If you are using the approach of a primary secondary, then you need to have also a control when you are using just a secondary antibody in the absence of the primary antibody. This will give you information about the non-specific binding of the secondary antibodies. And this actually leads me to another very important thing, which I forgot to mention, is that when you use primary cells, it depends which primary cells you're using. Some primary cells contain Fc receptors. And as it is saying, Fc receptors such as macrophages, dendritic cells, I have plenty of those. So they can bind your Fc portion of your antibody. So in this case, you need to do an additional blocking step. There are multiple reagents. We can use human serum, because if you're using primary cells from human, you can use actually Fc blocks. They already exist. And then you can use your primary antibody. Because even though you are using primary cells from human origin, and you are using to label them with or target your protein with mouse antibodies, primaries, they could be a cross-reactivity. So sometimes the signal what you are getting is maybe not a signal from your real protein, but it's just an attachment of non-specific of the Fc part of your mouse antibody to the Fc receptor on the surface of your primary cell. So this is a very important part of it when you are looking. Please make sure that you are having your cells do not express Fc receptors. If they express Fc receptors, please do an additional blocking step by human serum, for example, if it's a human.
This is very important, exactly. And Fc receptors, are they mostly in immune cells, or are there also other type of cells we need to watch out?
Yeah, you need to watch out, because in general, like T cells have a very low. Basically, they don't have Fc receptors, but NK cells, they have Fc receptors, like CD16, that's like Fc receptor. So in this case, you need to watch out. The best thing is to, if you are not familiar with the cell line, what you're using, or whether it has Fc receptor, please go to literature, have a look, and you will definitely find this information. On the other hand, you can always do Fc block. It's not harming. So if you include another half an hour incubation blocking step with the human serum, it's not harming for your staining. But at least then you'll be much, I will feel much confident that I don't have any Fc receptor in the surface.
So would you say, is it also useful to use higher blocking or blocking with serum in cell lines, or that doesn't really matter so much?
So you can have cell lines like a Th1 cell line, which is a macrophage cell line, which will have Fc receptors. Right. So I recommend that. So I will just double check whether my cell line contains Fc receptors.
Moving on to another application, we have a question regarding the visualization of synaptic structures in brain tissue. And the question is, if you can comment, or if you know what strategies are employed to optimize the labeling of synaptic proteins.
Yeah, so we have some applications and on our webpage that show how to do imaging in tissues. Yes, you can study imaging in tissues. What you need to make sure is that you have a very thin tissue slice. And then the strategy for labeling in tissue is exactly the same as is a strategy you will use for labeling a tissue for confocal imaging. But when you are going to do the imaging, you will need to use the imaging buffer, which is compatible with the fluorescent dyes of the antibody that you're using. But make sure that you have a very thin slice of a tissue. Ideally, if you are putting your tissue to a cover glass, and then you make a staining from the top, I would recommend to put a thin cover slip on the top, like number 1, as I mentioned in the beginning, and then reverse it and image it that way. Because that's the best way, because your staining will depend how deep you can go. In a tissue, you might not be able to penetrate through the whole tissue compared to when you have cell lines which are more transparent. So in this case, ideally, I will recommend that you do a staining on the top, you put a cover slip on it, you flip it upside down, and you image that one, which means that it will image the top part of your tissue.
Okay, talking about tissue and very exciting applications, I want to dive a little bit deeper into our last section of today, which is we called precious samples. All samples are, of course, precious, and we want to make the best protocols to visualize in the best manners. But actually, how would I go ahead and image cells in suspension? Can I image in suspension, or what do I need to do for dSTORM?
No, so as we said before, for dSTORM, imaging in suspension is currently not possible because your cells will just move around. And as we mentioned, you need to make your cell adhere to the surface, you need to fix them, and then you perform staining. Which means that when we talk about precious samples, I'm referring to samples which you maybe get a small biopsy, you isolate some primary cells from it, you still need to split it for experiments by using flow cytometry, maybe some functional assays, and you want to have imaging. So maybe at the end, you end up with a number of cells like 1,000. You're not getting any more in millions, but maybe you get 1,000. So what is the best strategy to do that? So first of all, just prepare your samples normally as you'll prepare for flow cytometry. Then you can use the surfaces, what we just mentioned. So use a polyalysine, fibronectin, collagen, and so on. But as I mentioned previously, please optimize that on a cell line, because you don't want to lose your last thousands of cells which you would like to image. And when you optimize that, follow the same strategy. You put your cells, you let them adhere, you fix them, you label them with already optimized protocol, and then you can do the imaging. Also, what's very important here, some of the dishes, if you look on a Petri dish with a glass bottom will accommodate approximately need a half ml of a liquid. So in this case, you might not have half ml of 1,000 cells in half ml, it's too much. So you want to reduce that. So when you have it in a very small volumes, I recommend to use dishes which can accommodate minimum like 20, 30 microliters or even 10. So we have a dishes, we have a flow chambers which can accommodate 5 microliter of volume. So in this case, you use the probability that your cells will be adhered within a time frame what you usually do is very high. So I recommend to take dishes which can accommodate smaller volumes. And most importantly, optimize your protocol on some cell lines previously.
Very important optimization steps. And I am curious to hear if the audience has any precious samples they want to discuss with us today. Please leave your questions in the chat. And in the meanwhile, I want to ask you, how could I image bone marrow cells? I imagine these are very tricky cells to image. And how would I go ahead, prepare a sample for dSTORM?
Prepare sample from bone marrow. I will actually just follow the same protocol what you use for any of your other assays. The difference between the imaging for a dSTORM imaging or even like for a confocal imaging is just to make sure that your cells are adhered to the surface. And the best way, or a more like a non-invasive way in this case to adhere them, is using a polyalysine as a straightforward first approach. You can use any other extracellular matrices if you want, but I would not really change the way of preparation yourself. So as you isolate your cells from bone marrow and you prepare them for FACS, please do the same thing. For flow cytometry, if you prepare them, just take the same samples, place them on a dish which has a polyalysine coating, and wait 15-20 minutes. You can look on the dish in a tissue culture microscope and see whether your cells are already settled. And if they are settled, just follow the fixation protocol. Which fixation to use will actually depend on your protein of interest. You will need to check it out. There's the two main ones, as I mentioned earlier, is a PFA, paraformaldehyde, or paraformaldehyde and glutaraldehyde combination.
Yeah. And I have a question regarding 2D and 3D models of cells. So the question goes, will my results differ in 2D or 3D cell models? Can you comment on that?
Yeah. So the answer in nutshell is yes. The reason is because when you do a 2D models, we don't have all this extracellular matrix. And we all know that if you look for a tumor environment or any environmental cell, it's not only our cell alone there. So what's in the surrounding will influence the behavior of your cell. And that's why when you are doing a 2D imaging, or 2D in general, 2D assays, they sometimes might not reflect the 3D assay. And in this case, if you want to do like 3D, like organoids, for example, you can do that. However, it will be very difficult to image them with a single microlocalization microscopy, because even if you will apply a 3D imaging of it, the 3D imaging is the depth is around 1 micron. So it will be very, very hard to image the full organoid. But yeah, there is a difference if you are imaging the behavior of cells or organization of receptors in a 2D setting might differ from the 3D setting. However, you can use gel matrices, extracellular matrix, the different combinations nowadays which can mimic the extracellular matrix, and you can include there in your solution some cytokines which can affect the behavior of your cells, in this case if you are talking about immune cells.
Talking about other precious samples, we have a question from Max. Hi, Max. So they grow cells on spider silk, and imaging the silk fibers is quite challenging. Do you have any tips regarding imaging silk fibers?
Wow, hearing it for the first time, but we did some fibers. We did a yeah. What I will suggest you that if you can actually grow your cells directly in dishes which already have a glass cover slip on the bottom, that will be a big benefit. Because in this case, if you need to manipulate with your cells afterwards and move them to a cover slips, you actually can break down this thing. So in this case, I would recommend that you are directly seeding and growing your cells on the dishes what you will use for imaging. In this case, I will recommend all the Petri dishes with a glass cover slip on the bottom, but then you will need quite a lot of volume to label it. However, you can also use some different 6 or 8 well dishes which has a glass bottom surface, but the minimum volume they need for incubation is like 100 microliter. So that's how I will suggest you to do that.
And then we have another question from the audience, and it's regarding imaging and labeling. So how can we enhance localization precision, particularly when dealing with sparse labeling in limited sample quantities?
Okay, so your localization precision will depend on your fluorophore, and that's affected by the laser power and the exposure time. So it's all dependent on signal to noise ratio. So, and that's independent on the structure you are looking for, because you are actually imaging the dye. So even if your sample is like under labeled or you are having a low expression, I would suggest that you characterize your dye within the imaging buffer on a structure where it's more visible, such as like mitochondria, tubulin, when you can see whether a structure was built up, and optimize there your signal to noise ratio. So to achieve better signal to noise ratio, you will need to most probably increase your laser power, maybe change your turf angle depending if you are in a perfect turf or you are in inclined illumination. And but it's dye dependent. So if you optimize your settings for imaging on a dye, for a specific dye what you're interested in to use, on a known structure, such as it could be even nuclear pores, can it be mitochondria, tubulin, then just apply the same settings on your other sample, which is more sparse. You will get a much realistic information about your sample. And don't worry if you don't get so many localizations. That's because you don't most probably have enough protein there. So that's a valuable information, but you know that your settings and imaging are determined for that dye perfectly previously on a known structure. That's what we recommend.
Right, and talking about little protein, actually, can you comment on the detection limit of, for example, flow cytometry compared to dSTORM?
Yeah, so the information is completely different. So if you are not using, let's say, so flow cytometry will give you information of individual cells, but you cannot see where the proteins are within a cell. If you are using image stream, then the resolution is very poor, because it's basically a snapshot of each cell what you are running through. It's a very good high throughput method which can use separate cells with whether you're expressing the protein or not. You can use sorting. But high resolution will give you an information about the position of your protein. So if you are, for example, let's say you're labeling a protein which is on a surface of I don't know, primary cells, let's say CD3, you might be all over. When you are using image stream as one of the imaging flow cytometers, you will just see a blob and it's there. But if you do an imaging of by using STORM, you will be able to see the individual receptors of the CD3. You will be able to identify whether you have more receptors per surface, let's say in a treated cell or untreated cell. So that's a different what this technology gives you. It gives you a spatial information of the protein. The flow cytometry gives you an overview of the protein. Is it there or not? Pretty much answer is yes or no.
I remember there was also a paper regarding, they were demonstrating they can detect low abundance protein in dSTORM, but it was not enough to detect in flow cytometry. So sometimes it can also.
Yeah, so this is possible because the number of localization what we are collecting, it's or depending on the fluorophore what you are using. Ideally, you will use a fluorophore like Alexa 4647, where usually all the imaging buffers are optimized for. And in this case, you can detect a low abundance of the protein by using STORM or dSTORM. But always you need to make a control. You need to make sure that if you put an isotype control of the same species, you are not detecting the same amount of localization, because if you do, then it's just a non-specific binding of your isotype control or your antibody to the surface and doesn't reflect it. However, if you see a complete significant difference between these two conditions, isotype control, whether my protein of interest is using antibody, then you can be confident that you are actually detecting the protein. And yes, I agree, you might not be able to detect in a flow cytometer because the sensitivity is different, much lower sensitivity.
Very good. I have one interesting question that we sometimes get from our users, and that is, can we actually also image plant cells with dSTORM? What do you think about plant cells?
That's a good question, but it's a little bit difficult. So if you're able to even isolate cells from the plant, we all know that they have a chlorophyll, right? So chlorophyll will absorb in a 488 channel, but also in a 640 channel. So any laser which you will use for activate your fluorophores will just increase the autofluorescence within a plant cell. So unfortunately, I would not really recommend to do STORM imaging in plant cells, as they are actually, you will get a lot of non-specific, like autofluorescent from the cell. Those problems.
That's a good thing to keep in mind. And are there also any specific human cell types that we need to watch out that have extremely high autofluorescence?
Yeah, so in terms of human cell lines, actually, I have a good example for that. So when I was working in immunology, we had some biopsies from COPD smoker patients, and they were isolating macrophages. And then what we actually realized is those macrophages are pretty black. And then when you shine a high laser, we use a different technology than just STORM. STORM actually worked very well with that, but in a different high resolution, actually it was STAD. I don't know, I think like we shine it high laser power. What happened? The cells just burst. And they burst because they were basically black. So they absorbed all the light, all the water inside the cell, right? And it just burst. So please watch out what you're imaging, because at some point you may get this really absorption of light inside the cells and just get it bursting. It was an interesting observation because I never saw anything like this before. So I would say like watch out here for it.
Me neither. I've never seen this. Were they were live cells or fixed cells?
Yeah, so well, they were live when we isolated them and then we fixed them. But even if you look for a bright field image just in a tissue culture, you already started to see that they were really black. Very interesting.
Wow. Okay. So another thing to watch out, the cell autofluorescence. Do not underestimate the effect and do not burst your cells, please. And with that anecdote, we already arrived at the finishing time of this webinar. And we would like you to thank everyone to attend our session today. Thank you so much. Don't forget that tomorrow we will still continue with a focus session on extracellular vesicle imaging. And on Friday, it will be a dedicated session for dSTORM analysis. So I'm looking forward to see you out there again tomorrow and on Friday. And thank you so much for your time, Stefan. Lovely conversation.
Thank you.
Bye-bye, everybody.